and thank you for listening. My name is Courtney Curtis, and I was a recipient of this year's Undergraduate Travel Award. Since this meeting is virtual, I hope I'm able to meet you all next year in person in Denver. I just graduated from Arizona State University, and during my last year there, I had the awesome opportunity to work on this project that I'll be telling you about today. This project began in 2015 and was intended to look at the effect of pesticide application in New Mexico to treat outbreaks of Mormon crickets. However, we were specifically examining the effects that the insecticide application had on the non-Mormon cricket insects. In this study, there were 12 sites sampled 3 times a year from 2015 to 2017. To obtain these estimates on the non-target organisms, there were 3 sampling methods: malaise, pitfalls, and sweep nets. There was a 2-week-long sampling period for malaise traps and 4 traps at each site. Because the 2015 and 2016 data had been largely collected and processed before I began working on this project, I will be focusing on the results from the 2017 field season only. Retrieval of these traps and the sweep net sampling occurred on June 20th, July 4th, and July 18th of 2017. Once the samples had been collected, I sorted them to class for all non-insect arthropods and order for all the insects. I then identified them to family using the 7th edition of Borer and DeLong. In addition to the identification process, I dried each order for 1 week before taking biomass recordings. The biomass results are not presented here, and neither are the results for any of the other orders. Since this is the Coleoptera Society, I will just be focusing on the families of Coleoptera that we found in 2017. So here are the families that we recorded over the 2017 field season, separated out by the trap type they occurred in. There were 9 families found in the malaise trap, 16 in the pitfalls, and only 7 in the sweep nets. However, there were unique taxa found in each of the different trap types, highlighted in red. Anthicity and Cantharity were only recovered in the malaise traps, Mordelity only in the sweeps, and 9 out of the 16 taxa in the pitfalls were unique to that trap method. These included some exciting finds like Ocodioides, the first I had ever seen, and a few wayward cerambicids that I wouldn't have expected to find in a pitfall trap. Next, we're going to look at the proportion of traps that had beetles in them. Please note that due to the way that the data was recorded in the previous years, we do not have abundance totals for these groups, just presence-absence information. For the next few slides, the blue wedges will denote the percentage of traps that included beetles, while the orange will represent the percentage that did not collect beetles. So here, for overall trap analysis, 3/4 or 75% of the samples had beetles in them. If we break this down by sampling type, we see that 64% of the malaise traps had at least 1 beetle present. And a similar result can be seen for the sweep nets, where 61% of the total sweeps had collected beetles. And then we have the pitfall traps. With a 100% catch rate for beetles, all 36 pitfall samples from 2017 had collected beetles, and anecdotally had the highest abundance of beetles overall. Now we're going to look at the number of traps each family occurred in. For these next few graphs, the x-axis is going to show the family names, and the y will have the number of traps that each family occurred in. Again, this is not abundance of each beetle family, just the presence absence in each trap method. From our earlier graph, 64% of the malaise traps had collected Coleoptera. So now with this sampling method, Coccinellity was the most commonly encountered family, being recovered in 14 out of the 23 traps that had beetles in them. The next most common was Mulierity in 8 traps and Clarity in 5. Next, for the sweep net samples, we have the same axes, again, the x being the families, the y being the number of samples that contained each family. In the sweep net samples, leaf beetles, Chrysonellity, were found in 13 of the 21 samples. With this method, we had Mulierity and Curculonidae as the next most commonly collected families. And lastly, here's a look at the pitfall data. This method had almost doubled the family diversity, and 100% of the samples contained Coleoptera. With this method, our most common families were, with very little surprise, Carabidae and Tenebrionidae. These families are extremely common in the habitats where the sampling was done and are often associated with the soil surface, making them ideal candidates for being collected in pitfall traps. There were some surprising results here as well. Staphylinity and Hysteridity were often collected in high abundance in pitfall traps, so we would have expected them to be in far more than just 1 trap each. And with that, I would like to thank the USDA APHIS Phoenix Lab Rangeland Unit for providing funding for this research project, and my supervisor, Derek Wohler. I would also like to thank the Arizona State University BioCollections for access to microscope, identification keys, and equipment. Thank you to Evan Waite, the PhD student who supervised this research. And lastly, I would like to thank Andrew Smith and Mark Branham for selecting me to receive this award and the Coleopterists Society for funding undergraduate student participation. Thank you so much, and I hope to see you all next year.